Good morning, students. So here once again we are back with the further stage, that is meiosis. Now, as we have discussed earlier about various stages of the meiosis, we are we have distributed entire meiosis process into two division. Its first division is meiosis 1, where it is going through prophase 1, metaphase 1, anaphase 1 and telophase 1. Second division is meiosis 2, which shows prophase 2, metaphase 2, anaphase 2 and telophase 2. Here in this diagram, if you see, the upper portion is showing meiosis 1 process and the lower diagram is showing meiosis 2. That was our last discussion about various stages of the meiosis. Now if I talk about today's session, today we are going to see various phases of meiosis 1 and that too prophase 1. So we are discussing now about prophase 1. Now if I talk about last discussion, what we have made is our requirement, that is multicellular organisms. We are actually sexually reproducing organism which are showing great number of the variation, that is either new structure or new function, and in that manner we are showing the huge complexity. Now in order to create the complexity, we are supposed to make change in features or characters. In turn we are supposed to make change into the protein. Now in order to make change in the protein, we are supposed to change our genes, that is our DNA, right? So that was the thing we are right now putting into the center of our discussion. So we are supposed to make changes into the gene. Now how we can change the gene, or which are the cellular process which is responsible for make changes into the gene, that's what we are supposed to see in prophase 1. So which are the necessary things which are taken care in order to create some variation or some changes into the genes. So if I talk about the meiosis is divided into meiosis 1 and meiosis 2. Now in meiosis 1, prophase 1 is further dividing into 5 steps, right? And these 5 steps, if we talk about, it is going to make all the necessary changes which are required. Look, if you think about first step, this is the first step which is known as Laptotene. Second is Zygotene. Third is Pachytene. Fourth is Diplotene. And fifth is Diakinesis. So these 5 steps are responsible for some necessary changes which has to happen in your DNA, and it is done by exchange of gene, right? The responsible phenomena which is happening here is exchange of genes. Now if I talk about the process, number 1 process, if you remember, is Laptotene, right? So in Laptotene, if you see, this is the process name, it is Laptotene, and there if you see, the chromosomes are formed from chromatine. Step number 2, that is zygotene. During zygotene, they are placed nearby. That is, they are arranging into, or they are actually getting arranged into two similar types of chromosome which are placed nearby to each other, right? So that is one special mechanism which is seen into meiosis. So here pairing of chromosome happening, right? We will see that later on, which are the things, what it is to be said, right? So that's what we are about to see later on. During the third phase, that is Pachytene, if you see here, they are putting each other parts over one of the arm of the other chromosome, and at this place they are changing their genes with each other. So genetic material in diplotene gets joined, and in diakinesis it is exchanged. So genetic material has to get exchanged, and that is where we can produce necessary variation or mixture of different types of characters altogether. So let us see stepwise each and every happening phases, that is Laptotene, Zygotene, Pachytene, Diplotene and Diakinesis. So if I talk about Laptotene phase, right? If I talk about the Laptotene phase, during Laptotene phase, only one thing which is responsible is chromatin fiber becomes longer and slender chromosomes. That is the only thing which is happening there. So prophase 1, during Laptotene, if I talk about first phase, that is Laptotene, the chromatin is converted into chromosomes. In the other word, I can say it is condensation of chromosomes, and this is the only mechanism which is happening during Laptotene phase. Only condensation of the chromosome and nothing else. Ready? So if you see the diagram here, you can clearly see that only one thing which is happening into the center, and that is condensation of the chromosome. Fine. So this is the first thing. Now due to the condensation of chromosome, nucleus is getting enlarged, becoming more larger. So these two important character of leptotene or leptonema, the stage is leptonema or leptotene stage, right? Please take the screenshot of this slide so that you can make a note easily. Fine. The second phase is zygotene. Now during zygotene there is very important phenomena which has to happen, and that phenomena is pairing of chromosome. that phenomena is going to happen, pairing of chromosome. Now, due to pairing, two types of chromosome are getting together. For example, if I talk about one pair is paternal pair, that is mentioned in black color, and second one is maternal pair, that is black, red in color. So they are actually arranged so nearby and parallelly, and that is why they it is considered as pair of homologous chromosomes. So this is actually happening during zygotene phase, pairing of homologous chromosomes. For example, the black one is chromosome number 1, but it is paternal, and the red one is chromosome number 1, but it is maternal. So they are coming together and make a pair. It is the speciality of meiosis process. Now, why it has to happen? Let us discuss the reason. Look, black one is having entire different set of chromosome, that is DNA, and red one is also having their own special type of DNA. So exchange of DNA must be during or between two different type of DNA, not similar type of DNA. So it has to happen between black colored DNA and red colored DNA. That is, now to be more specific, during or between paternal and maternal DNA. Not red one and red one, or black one, not between red one and red one, and or not between black one and black one, but it has to happen between red one and black one. And this is the reason why phenomena which is happening here is pairing of homologous chromosomes. So let us discuss why it is happening, because of exchange of genetic material of maternal DNA and paternal DNA, and that is how we can produce the variation. So to create variation amongst the offspring or next generation, during zygotene, pairing of homologous chromosome is happening. Ready? So this is why these phenomena or this phase is very important. So here, if you see the diagram, here synaptonemal complex forming, right, between two different, one is here, it is mentioned blue, and here the other is mentioned as the red one. So if you see into the detail of the zygotene, chromosomes become more condensed and similar chromosomes starts pairing together, and that pairing is termed as synapsis with the help of a complex structure called synaptonemal complex. You are supposed to remember, right? The structure which is responsible for pairing, that is synapsis, is synaptonemal complex. It is a joint, what we can say, right? It is a joint from where they are getting joined together. Now the pair of chromosomes are called homologous chromosomes. So it is happening during zygotene or zygonema. Each pair of homologous chromosome is called bivalent. right? Each one. So if I talk about this blue one, it is bivalent chromosome. And if I talk about this red one, it is also bivalent because it is having two arms, one and two, one and two. Ready? So each synaptonymal complex which is joining these two pair of chromosome, two pair of homologous chromosome, after joining it is called pair of homologous chromosome. So this is number 1 homologous chromosome and this is number 2 homologous chromosome, and both are bivalent and both are getting together and joining with the synaptonymal complex, right? And that is how they are forming as all the chromosome number 1, chromosome number 2, chromosome number 3, chromosome number 4, 23 pair of chromosome will be forming different pair of homologous chromosome in our cell when they are dividing with the meiosis process. That's what you are supposed to remember. Fine. So it is called pairing of homologous chromosome, which is happening during the Zygotene phase. So please take the screenshot to make a further note. Now comes the phase number 3, that is Pachytene. Ready? Pachytene. It is the next step of the Zygotene. Now why it is next step? Because the synaptonymal complex which is adjoined, right, it becomes more complex. Complex joint, right? It is getting more complex, it is joining with more stable structure, and from the center towards their peripheral region, if you see, it is joined with perfect manner with a specific type of protein in between. So this is making more stable pair between two homologous chromosome, right? So in this phase, joint is become more stable from the point of their center. In pattern of zipper, I repeat, with the type of zipper, from center to peripheral region it is getting joined. That's what you are supposed to remember. So zipper pattern joining. Why this is happening? Because these complex joint must be proper enough so that this blue chromosome and these red chromosomes gets perfectly joined at the junction. So it has to get settled very nicely for its proper joining, and that is how the entire structure is made. So comparatively it is most longer phase. Why? Because it takes so long time, and bivalent chromosome splits into similar chromatid, and this stage is called tetrad. Look, bivalent chromosomes, they splits into similar chromatids. Now if I talk about this phase, for example, this is chromosome number 1, homologous chromosome number 1. This is homologous chromosome number 2. Now this one is synaptonymal complex, from where it is getting joined, but after joining from centromere part to their respective peripheral region, they are getting joined together. So actually from center 1, 2 and 3, 4. So this is chromatid number 1, this is chromatid number 2, this is chromatid number 3, and this is chromatid number 4. As it is joined from the centromere with a specific protein, so it is unit or single centromere, it becomes single centromere, and it is producing these 4 separate chromatid from one centromere, and that is why this phase is known as tetrad. Ready? So tetrad formation is its speciality. If I talk about this zygotene phase, it is having by 2 bivalent chromosome which are joined. But from centromere they still are not joined, but they have joined at the synaptonymal complex. Ready? Now if I talk about the third phase, right? If you see into the diagram, they are joined with the centromere. So let us consider that part in parenchyma. Pachytene is the next stage, where if you see pairing a chromosome here is becoming more complex. So complex pairing. Now second important phenomena which is happening, becoming tetrad. Why tetrad formation? Because they are they matlab both the bivalent chromosomes joined together from centromere, and that is why it is becoming tetrad. So that's the speciality of Pachytene. Ready? Appearance of recombination nodule at crossing over site. Recombination nodule is here. This part is called recombination nodule, and it leads to recombination of genetic material. Exchange of gene is happening there. So recombination is completely by end of Pachytene, right? Recombination completed by the end of Pachytene. So why this tetrad formation or complex process happens is because if two non sister chromatid joins together, then exchange of genetic material between, if I talk about non-sister, that is maternal and paternal, is possible. So there two non-sister chromatids are joining together, pairing together, and that is why it was previously the combination of gene. Right now, exchange of gene is happening between these two non-sister chromatid, and that is why it is called that is again combination. So a recombination is the result of exchange of gene between two non-sister chromatids, and this is a very special mechanism or speciality of this Pachytene phase. Ready? So recombination happens during Pachytene. Recombination nodule, that is also a speciality we must not forget to write. Recombination nodule appears. It is the site of recombination. Recombination nodule is a site of recombination. Ready? So that's the speciality of the Pachytene. So recombination happens during the Pachytene, and genetic material it is getting exchange between maternal chromosome and paternal chromosome. That is why a mixture of two generation or two different types of individuals are seen into the next generation of the offspring. Ready? So that's the speciality of Pachytene. So please take the screenshot so that you can make a further note. Now comes the Pachytene. Okay, this is what we have already discussed. Diplotene, the next stage number 4, diplotene. Fine. During the process of exchange of gene, when recombination nodule is perfectly joined, I repeat, where recombination nodule is perfectly joined, you can see the tetrad which is happened, right? You can see the tetrad which is happening. Due to the tetrad, we have seen this is tetrad. We have seen that from the center or through the centromere, joining of these chromosomes towards their peripheral region appeared. So in between they have special type of joining, right? Now that has to be dissolved, and both the chromosomes are again to be separated, and that's the speciality of diplotene. It is getting the separation of two chromosome again. Ready? So if you see the speciality, so diplotene is dissolution of the synaptonymal complex which was happened in the Pachytene, right? So during Pachytene, whatsoever join between two chromosomes have complexly made is here dissolved. So dissolution of the synaptonymal complex occurs and a recombined homologous chromosome of again bivalent separated. So tetrad was again separated into two bivalent chromosomes. So that is a speciality which is shown by Diplotene. If you see the name Diplo, that is two. So it is separation. Meaning is separation of, look, recombined homologous chromosomes. It is the separation of recombined homologous chromosomes. We are making it apart from each other, so that from tetrad back to two bivalent homologous chromosomes. So that's the result. From tetrad you get two bivalent chromosome back into the action. Now if you see, due to this separation, if you see the diagram 1, 2, 3 and 4, X-shaped structure you can see between the site of recombination between two non-sister chromosomes. This X-shaped structure is called chiasmata, right? So chiasmata appeared between two non-sister chromosomes at the site of the recombination. So that's the speciality of this phase, Diplotene. Chiasmata. It is the X-shaped structure appeared or appears between two non-sister chromosome, non-sister chromatid at the site of recombination. So that's a very special event which is happening during phase of Diplotene. Ready? Now in oocytes of some vertebrate, Diplotene lasts for months or years. That long it can last. That's the thing you are supposed to remember. Otherwise it is a short gap or short type of the phase. Ready? So please take the screenshot of the slide so that you can remember to note down. And the last one is diakinesis. In diakinesis, if you see, the chromosomes are completely set apart from each other and settle down for further process. Ready? So main event which is happening is terminalization of chiasmata. What is terminalization of the chiasmata is only the place of the chiasmata chromosomes are joined Otherwise, all chromatids are separated. Except site of chiasmata, and that is why it is called terminalization of the chiasmata. This phenomena is known as terminalization of chiasmata. So that's the speciality of diakinesis, the fifth phase. Dia, that is to, kinesis, that is movement. So they are getting apart from each other. They are getting separated from each other completely, but except one place, right? So complete separation of chromosomes except site of chiasmata, and that is called terminalization of chiasmata. So that's the speciality of diakinesis phase. Chromosomes are fully condensed as it was, and mitotic spindle fibers originates from poles to prepare the homologous chromosomes for separation, right? So prophase is actually also responsible for production of mitotic spindle. Here meiotic spindle are produced, but in the diakinesis phase, nucleolus, nuclear envelope disappears here. So rest of the things which are to be happen during prophase has to happen in diakinesis. So spindle formation, nuclear envelope dissolution, and nucleolus disappears. So these are the changes happening into the diakinesis, and that's how prophase 1 is ended. So all the necessary steps which are left for prophase are completed into the diakinesis. So it is ready for the metaphase. So prophase 1 is ending at the two separate chromosomes, mitotic spindle, nucleolus and nuclear envelope disappearance, and that is why we can say that entire prophase is getting ended at the diakinesis stage. Now cell is ready for the metaphase 1. So we are starting with metaphase 1 into the next discussion, into the next lecture. Till then, have a nice time and bye.